Okay guys, so today we're going to do a blood typing case. So if you have watched my previous video, you already have learned how to do the typical blood typing. So here is basically the experiment you guys would be doing normally in the class. So I'm gonna run the experiment for you, and you guys, based on the data you're observing in this, you can answer the question and complete the chart for the blood typing sample. So here what I have is 4 blood samples. Here is, if you read this, it says Mr. Green sample, blood sample. This one says Ms. Brown. We have a sample for Mr. Jones, and then another sample for Mr. Smith. Basically what this means is we have 4 samples from 4 people, and we have no idea what this blood type is going to be. So what we're going to do is, like, as you can notice, we have 4 trays, and we're going to try to use these trays and our original blood type and what you have here to identify the blood type. So right here you have 3 different solutions. So if you read the label, and I don't know if you can see them from the thing, it says simulated anti-A serum. What that means is basically this is a man-made anti-A antibody. This one, it says a simulated antibody B serum, which means this is a man-made anti-B antibody. And the last one is for RH, again simulated anti-RH serum, which means this is anti-RH antibody. Again, these are not what we can find normally in the human body, but these are what we can use to identify what is an individual's blood sample is if we have no idea what their blood type is. So what I'm going to start with is Mr. Green blood sample. So I have these trays. Move this one upside down. If you can see in the video, there is a letter A up here. There is a letter B down here, and there is letter RH right here. And it's present in all of our trays. It's basically identical. So I want to see if Mr. Green has anti-A, or basically, antigen A, antigen B, or antigen RH present on their blood. So I'm gonna add a couple drops of blood sample to one tray. They're all, again, notice they're from Mr. Green. And the one that has the label A, I'm going to add the serum anti-A to it. So again, I'm gonna add a couple of drops to that. To the one that has the letter B, I'm gonna add my anti-B serum. So here's my anti-B serum. And again, lastly, to the one that is the RH, I'm gonna add the RH one. So here it is. And what I'm going to do is now I'm gonna mix my sample. So understand that when I'm mixing the sample, you don't wanna cross-contaminate. So if I'm using my toothpicks to mix this sample right here, I don't want to transfer this to the other sample. So I'm going to use a different toothpick and mix the other sample. And again, repeat it again for my B portion. So again, in these scenarios, anytime I'm creating a blood sample, I'm looking for any form of clumping, anything that is different from what I originally started with. And if you look at this sample, I don't know how clearly you can see this. You can see anything in this sample. This is pretty clear still. This is also pretty clear, but notice this one. You see there's a lot of bumps in there, little circles in there. This means that there is a reaction in this one. So that's identified as clumping or agglutination. And actually, if I'm honest, you also see reaction in this one. Again, notice how there is that clearness is gone away, and now you see a lot of structures forming in here. So this is also identified as a reaction. So for Mr. Green, the reaction was observed in RH and was reaction observed in B. Now let's go ahead and repeat this experiment with Ms. Brown. Again, I'm gonna do the same steps. So a couple of drops of blood sample to all 3 wells, and then match the corresponding anti-serum to each of these. So I have my B, I have A, oops, And then I have my RH. And again, what I'm looking for is anything outside what I actually started with, which was a clear blood sample, which is what I started with. So I'm gonna mix this sample again. So this is one sample. This is another one. I'm gonna spread it as much as I can so you guys can see if there is anything. And then this is the third one. So looking at this, take a second to take a look. So if you are looking at this, again, take a look, and based on what you're observing, you should be able to draw your own conclusion. So I'm gonna repeat this again for the next sample, which is Mr. Jones. I guess there's bottle leakage in this bottle. And these are all fake samples of blood, so there is nothing that in terms of getting, transmitted any type of blood disease with it. So you don't have to worry about this if you were doing this in class or in person. Same process repeated. So here is one sample. Here is another one. And here is our third sample. So again, we're looking at any significant changes in our blood sample. So let's take a closer look. So here is our first sample. Sorry guys. Here is our first sample. Again, look in this sample, you have kind of a cloudiness in the sample. And then on the top one, you can see some small structures being formed. And then lastly, we're going to do our last sample, which is Mr. Smith. Same process over. And by the way, I will make the pictures of these slides, this presentation also available. So if you guys want to take a closer look yourself, you're welcome to do so. So here's our first sample. Nothing is visible happening in there. Here is our second one. There is definitely something happening in there. So you can see a lot of things are forming in there, right? It's almost solidified. And then here is our third blood sample. No visible change here. So I just wanted to take a moment and actually go back to the first sample we did, because now that it's been left alone, we saw changes in the first sample we did. We saw changes in B and RH, and now that it's sat for a bit, we're also seeing changes in A as well. So guys, here is your lab results. You guys go ahead and take a look at this, and based on this data, you can make your own conclusion as far as where you see the changes happening, okay? And again, look for the pictures in your files for a copy of the video pictures that I'm showing you right now.